Welcome to the deep dive, or as we're calling it today, Talk Tensor's your shortcut to some really interesting machine learning research.
Hey there.
We've got a paper today that, well, really caught my eye. It's about using deep learning for potentially detecting Alzheimer's disease super early.
Yeah, and the key here is how they're doing it. They're looking at detailed scans of the back of the eye, the retina, using something called OCT.
OCT, right, optical coherence tomography. And the idea is that AI, specifically deep learning models, might be able to see subtle signs of Alzheimer's in these eye scans, maybe years before someone even shows symptoms.
Exactly. And that's crucial because, Alzheimer's has this really long preclinical phase. We're talking maybe 15, sometimes even 20 years before memory problems become obvious.
Wow. So by the time you notice something's wrong, the disease is already quite advanced.
Precisely. And the current ways to diagnose it definitively, things like analyzing spinal fluid or doing specialized PE brain scans, they're not exactly easy. They can be invasive or expensive or just take a lot of time.
Which is why this idea of using retinal scans is so appealing, right? It's non-invasive, relatively quick, potentially much cheaper.
That's the hope. The paper really digs into this connection between the eye and the brain. The retina isn't just, lining at the back of your eye. It's actual neural tissue.
Like an extension of the brain, you can see.
Kinda, yeah. Embryologically, they develop together. And OCT lets us see the different layers of this tissue in incredible detail. Think of it almost like a high-res optical ultrasound for the retina.
And the thinking is, changes happening in the brain because of Alzheimer's might actually show up as changes in these retinal layers.
There's growing evidence for that. Studies have shown links between AD and visual problems, like trouble with contrast sensitivity or visual memory, even early on.
Okay, so it's not just theoretical.
Not at all. Researchers have found amyloid beta deposits. Those are the sticky plaques famous in Alzheimer's in the retinas of AD mouse models. And they've seen similar things, plus neurofibrillary tangles in the visual areas of human brains with AD.
And OCT scans in people with AD. Have they shown differences?
Yes, several studies have reported thinning, particularly in the retinal nerve fiber layer, the RNFL, in patients with Alzheimer's compared to healthy individuals.
RNFL, retinal nerve fiber layer. Got it.
And this isn't just in full-blown AD. Similar, though maybe more subtle, thinning has been seen in people with mild cognitive impairment, or MCI, which is often considered a very early stage of AD.
So the retina might be like an early warning system.
Potentially, yes. Post-mortem studies have confirmed the presence of those amyloid plaques and tangles in the retinas of AD patients. It's not unique to AD, though other neurodegenerative diseases like Parkinson's and MS can also show retinal changes, which is an important caveat.
Right, so it's not a simple one-to-one link, but still a really promising avenue.
Definitely. Some studies suggest specific measurements, like the thickness of certain parts of the RNFL, could be biomarkers, but maybe not sensitive enough for the very earliest stages on their own. That's where machine learning comes in.
Ah, okay. Because the changes might be too subtle for the human eye or simple measurements to catch reliably?
Exactly. The subtle patterns across the entire scan, the texture, the relationships between layers, that's what deep learning algorithms are incredibly good at finding. They can analyze these complex OCT images or even OCT angiography, which looks at blood vessels
OCTA?
or standard fundus photos, and potentially pick up on disease signatures that we might miss. There's already been work using machine learning on OCTA data, for example, for classifying AD and MCI.
And this paper specifically dives into using deep learning on the standard OCT structural scans.
Yes, they used a public data set called Dryad and evaluated a whole suite of deep learning models, especially focusing on transformer architectures, to see how well they could distinguish between OCT scans from AD patients and healthy controls, the CO group.
Okay, let's dig into their methods. What do they actually do?
Well, the main goal was clear. Build a robust AI tool using these OCT scans for early AD detection. First step, obviously, was data preparation.
The Dryad dataset you mentioned.
Right. It has OCT scans sorted into folders for AD subjects and control subjects. They developed a specific process to load this data, resize the images to a standard size 224 by 224 pixels, common for many pre-trained models.
Why resize? Does the original size vary a lot?
It can, depending on the exact scan protocol. Standardizing makes it easier for the models. They also normalize the pixel values, another standard step, basically putting all the images on a similar brightness and contrast scale.
Makes sense, like adjusting photos before comparing them.
Exactly. They mentioned the original scans were taken with a Heidelberg Spectralis OCT machine using specific settings like a 30-degree field of view and averaging multiple scans up to 100 to get cleaner images.
Averaging helps reduce noise, right?
Yes, precisely. Then they used software called ImageJ for further pre-processing things like noise reduction again, brightness and contrast tweaks, extracting the raw image data, cropping unnecessary parts, and resampling to make sure the pixels represented equal distances horizontally and vertically.
So quite a bit of careful image cleanup before feeding it to the AI.
Absolutely essential for good results in medical imaging AI. After all that, they had standardized image files like PNGs or JPGs ready for the models. They set up an 80-20 split, 80% of the data for training the models, 20% held back for validation to test how well they learned.
Okay, and then the models themselves, you said they tried a range?
They did. They started with a simple custom CNN, just to get a baseline, I suppose. Then they moved to well-established architectures like VGG16 and ResNet 50.
ResNet is the one with those residual connections that help train really deep networks, isn't it?
That's the one. Then they tried Inception v3, known for using parallel processing paths, EfficientNet B0, which smartly scales up network depth, width, and resolution.
EfficientNet, yeah, that made a splash.
DenseNet 121, where layers get inputs from all preceding layers, which is great for feature reuse, and NASNet Mobile, a model actually designed by another AI through neural architecture search.
Wow, okay, so a real tour of modern CNNs.
Right.
But the focus was on transformers.
Right. They used a pre-trained Swin transformer, which is really state-of-the-art for vision tasks. It builds a hierarchical representation, kind of like CNNs, but uses attention mechanisms, like transformers in language, to relate different image patches.
Attention.
Exactly.
So it can focus on the most relevant parts.
In a sense, yes. It learns relationships between different parts of the image, even distant ones. And then there was TransNet OCT.
The other big one mentioned in the intro.
Yeah, TransNet OCT is particularly interesting because it's specifically designed for OCT. It combines transformers and CNNs, aiming to be really good at segmenting the different retinal layers within the scan, not just classifying the whole image.
Segmenting, meaning identifying the boundaries of each layer, like the RNFL we talked about.
Exactly. And they use specific metrics to evaluate how well TransNet OCT did the segmentation, things like ASSD, DSC, HD, alongside the usual classification accuracy.
Okay, so they trained all these models using the 80% training data, aiming to minimize prediction errors using standard techniques like cross-entropy loss and the atom optimizer.
Over multiple passes through the data, called epochs. Then they tested them on that held-aside 20% validation set.
I measured things like accuracy, precision, recall, F1 score, but the usual suspects for classification tasks.
Right. They also generated confusion matrices, which basically show you what kind of mistakes the model made, like how many AD cases were mistaken for controls and vice versa.
And you mentioned CAM earlier, class activation mapping.
Yes. This was crucial, especially for the Swin transformer and TransNet OCT. CAM produces a heat map overlay on the original image, showing which pixels or regions the model paid most attention to when making its decision.
So you get a visual clue about the model's reasoning.
Exactly. Is it looking at the optic nerve head? Is it focusing on thinning in the ganglion cell layer, or is it just latching on to some weird artifact? It's vital for building trust and understanding, especially in medicine.
Makes sense. You wouldn't want a diagnosis based on the AI focusing on a smudge on the lens.
Precisely. They also used 5-fold cross-validation, particularly for the Swin transformer and TransNet OCT. This means they split the data 5 different ways, trained and tested 5 separate times, and averaged the results. It gives you a much more robust estimate of performance and how well the model might generalize to new, unseen data.
So more rigorous than just a single 80-20 split.
Definitely. And for TransNet OCT, they also mentioned using data augmentation during training, things like slightly rotating or flipping the images to help the model become more robust to variations. And they used a combination of loss functions, including dice loss, which is often good for segmentation tasks.
Okay, so that's a pretty thorough methodology. What were the results? Did the fancy models work?
Oh, they did. The training curve showed what you'd expect, loss going down, accuracy going up over the epochs, but the comparison table was the real headline.
Let's hear it.
Well, the simpler CNNs were okay, but the more advanced ones did better. DenseNet 121 performed quite well. But the Swin transformer really stood out among the pure CNN-like architectures, hitting over 93% accuracy on the validation set.
93%, that's pretty good for distinguishing AD from controls just based on an eye scan.
It is. The confusion matrix for Swin showed it correctly identified most cases, with only a few misclassifications, a couple of AD cases called controls, and a couple of controls called AD.
And the cross-validation held up.
Yeah, the five-fold cross-validation average accuracy for Swin was also around 93.45%, so it seemed pretty robust.
And the CAM visualizations for Swin, what do they show?
That was interesting. For correctly classified AD images, the CAM maps often highlighted areas consistent with known AD pathology, like thinning of the RNFL and the ganglion cell layer complex. For healthy controls, it often focused on areas showing thicker layers, or sometimes the choroid beneath the retina.
So it seemed to be looking at biologically relevant features.
Generally, yes. Although, when they looked at misclassified images, the CAM could sometimes explain the error. For example, an AD case misclassified as healthy might show thicker than expected RNFL in the highlighted region.
Okay, what about TransNet OCT? You said it did even better.
Significantly better. On the original unsegmented OCT images, TransNet OCT achieved an accuracy of 98.18%.
Wow, nearly perfect.
Yeah, incredibly high. Precision, recall, F1 score were all around 0.98, very low validation loss.
And did they also run Swin and TransNet OCT on segmented images where the layers were already outlined?
They did. Interestingly, the Swin transformer's accuracy dropped a bit on the segmented images, down to about 89.8%.
Lower. Why would that be?
The author speculated that maybe the segmentation process, while highlighting key layers, might inadvertently discard some subtle textural information or context from the full image that the Swin model was actually using effectively on the raw data.
Huh. So sometimes giving the AI less processed data is better.
It seems counterintuitive, but possibly, yeah. The model might find useful patterns in the raw appearance that get lost when you just give it clean boundary lines.
What about TransNet OCT on segmented images? It's designed for that, right?
It is, and its performance went up even further on the segmented data, hitting 98.91% accuracy.
Okay, so TransNet OCT excels with that layer information explicitly provided.
Seems so. Its segmentation metrics were also very good, low error, high overlap between predicted and actual layers. Though the Hausdorff distance, HD, was infinite, suggesting maybe some outlier boundary prediction issues occasionally. But overall, very strong performance.
And the CAM maps for TransNet OCT.
They also showed the model focusing on relevant areas, often highlighting specific boundaries between retinal layers, which makes sense given its segmentation capabilities. It seemed quite targeted.
So overall, the takeaway is that these advanced deep learning models, especially transformers and hybrid approaches like TransNet OCT, are really effective at this task.
Absolutely. They significantly outperform simpler CNNs. The paper emphasizes the power of these pre-trained, sophisticated architectures. The interpretability from CAM is crucial, though they noted that while TransNet OCT seemed to consistently highlight relevant areas, the Swin transformers CAM maps were sometimes a bit less clearly focused, possibly needing more data to really nail down consistent patterns.
Which points towards future work, I guess.
Exactly. Larger data sets are always needed in deep learning, especially medical imaging. Higher resolution scans might help too. Exploring multimodal inputs, maybe combining OCT with OCTA or fundus photos could be powerful. And testing even newer architectures is always on the table.
And they mentioned the caveat about other diseases causing similar retinal changes.
Yes, a crucial point. Non-neurologic eye diseases or even other neurodegenerative conditions can sometimes mimic these patterns. That really highlights the need for large, diverse, and longitudinal data sets following people over time to really sort out the specific signatures of AD.
Okay, let's zoom out a bit. How does this fit into the bigger picture of Alzheimer's research and diagnosis?
Well, for decades, AD research relied heavily on examining brains after death. Then came the ability to measure biomarkers like amyloid and tau in cerebrospinal fluid CSF via spinal taps.
Invasive, though.
Right. And then came PET scans that could visualize amyloid plaques and later tau tangles in the living brain. Huge breakthroughs, but PT is expensive and not widely accessible. MRI can show brain atrophy, but often later in the disease.
So there's always been this push for something easier, cheaper, less invasive.
Exactly. And the idea of the eye as a window to the brain isn't new in medicine generally. Ophthalmologists have long been able to spot signs of systemic diseases like diabetes or high blood pressure just by looking at the retina.
So applying that to neurological disease makes sense.
It does. The development of OCT technology in the 90s and its refinement since then gave us the tool to see the retina's structure non-invasively and in such detail. Then, maybe in the last 10, 15 years, deep learning exploded, especially in image analysis.
The ImageNet moment and all that.
Pretty much. Suddenly, we had algorithms capable of learning really complex patterns from images, perfect for medical scans, and more recently, transformer models, which revolutionized language processing by understanding context, were adapted for vision.
Like the Swin transformer they used.
Exactly. So this paper sits right at the intersection of these advancements. High-res retinal imaging meets powerful deep learning, specifically transformers, applied to a major unmet need, early AD detection.
Okay, so distilling the core points here. Retinal OCT is non-invasive and shows promise for early AD screening.
Check.
Advanced deep learning, particularly models like TransNet OCT and Swin transformer, achieve very high accuracy in telling AV scans from healthy ones.
Check. Seems like transformers have a real edge here.
How you prepare the images, like segmenting layers, matters and might affect different models differently.
Yep, that was an interesting finding. Maybe raw data is sometimes better.
And using tools like CAM to understand why the AI makes a prediction is super important for trusting and improving these systems.
Absolutely critical for clinical translation. You need that interpretability.
And the big picture implication. This could genuinely change how we diagnose and manage Alzheimer's.
That's the potential. Earlier diagnosis means earlier access to whatever support and future treatments become available. It could make screening feasible for wider populations, not just those already showing symptoms or with strong family history.
Maybe even help monitor the disease or response to treatment over time.
That's another huge potential application, yes. Plus, understanding which specific retinal features are changing might give us new clues about how the disease actually works at a biological level.
All right, this is really exciting stuff. It leads perfectly into our novel notions segment. Where could this go next?
Okay, so building on the high sensitivity, especially of TransNet OCT, one idea is a truly long-term longitudinal study.
Longitudinal following people over time.
Exactly. Imagine identifying people in their, say, 30s or 40s who have a high genetic risk for Alzheimer's. Maybe they carry the APOE4 gene variant, for instance.
Okay, high-risk group.
And you start performing regular OCT scans on them, maybe annually, analyzed by something like TransNet OCT. You do this for decades before any cognitive symptoms are expected.
The goal being to catch the absolute earliest signs of change in the retina.
Precisely. The hypothesis is that these powerful models could detect subtle progressive changes years, maybe even a decade or more, before current clinical tests or even CSF or PET biomarkers turn positive. We'd be mapping the disease's retinal footprint from its very inception.
Wow. The impact there could be massive, couldn't it? If you could identify someone on that trajectory 15 years before symptoms.
It could revolutionize prevention. Imagine starting protective therapies or interventions that early. It might dramatically slow or even halt progression for some. Plus, the patterns you discover in those early retinal changes could point to brand new therapeutic targets we don't even know about now.
What's the catch? Why isn't this already happening everywhere?
Well, the main hurdles are practical. It requires immense long-term funding, incredible participant commitment over decades, and careful management to ensure the data is consistent and the cohort remains representative. It's a huge undertaking.
So, probability. Moderate, maybe. High potential reward, but high logistical barriers.
I think that's fair. Moderate probability, but potentially transformative if successful.
Okay, here's another angle. AD doesn't just affect vision, right? Early on, other subtle things can change, like sense of smell, maybe even tiny changes in how someone walks.
That's true. Olfactory dysfunction is a known early sign, and changes in gait or motor control can also occur, often very subtly at first.
So what if you combined retinal OCT data with other non-invasive sensory or motor data, like standardized smell tests and maybe data from wearable sensors that track gait patterns, stride length, balance, that kind of thing?
Ah, a multimodal approach. Interesting.
Yeah. You feed all this diverse data, the OCT images, the smell test scores, the gait parameters from wearables, into a sophisticated AI model, maybe a transformer designed specifically to handle these different data types and find connections between them.
The rationale being that integrating subtle signs from multiple affected systems, visual, olfactory, motor, might give you a stronger, more reliable signal of early AD than any single one alone.
Exactly. A more holistic assessment. You might catch people earlier or with greater certainty by combining these weaker individual signals. Transformer networks are good at finding those kinds of long-range dependencies across different inputs.
The potential real-world impact there seems quite high in terms of accessibility. Smell tests are easy, wearables are becoming ubiquitous. Combine that with a relatively quick OCT scan.
You could have a low-cost, multi-pronged screening tool potentially suitable for routine checkups, much more accessible than, say, PE scans or spinal taps for initial screening.
Right. The missing piece currently is often this integration. We tend to look at biomarkers in isolation. The challenge would be the data fusion, how best to combine these disparate data types, and of course, rigorous validation. But the probability feels, well, quite high, actually. Sensor tech and multimodal AI are booming.
That feels like a really powerful direction, combining these different non-invasive windows into neurological health.
It does. It really moves towards a more systems-level view of detecting the disease early.
Well, this has been absolutely fascinating. This paper really underscores the incredible potential at the intersection of advanced AI and medical imaging, particularly with retinal OCT for Alzheimer's. If you want to dive deeper, definitely check out the paper links in the description.
Yeah, it's impressive work. And it really makes you wonder, doesn't it, as these non-invasive AI-driven tools get better and better, how fundamentally will they change our approach to tackling not just Alzheimer's, but maybe a whole range of neurological conditions in the coming years?
A powerful thought to end on. Thanks for joining us for this deep dive on Talk Tensors.